Thank you to everyone for joining us, and thank you so much to our presenters. I'm Monica Coonrads, founder and CEO of the Rett Syndrome Research Trust, and mother to a 24-year-old young woman who was diagnosed with Rett syndrome at the age of 2. I'm joined today by two of my colleagues, Dr. Randy Carpenter, our Chief Medical Officer, and Dr. Jana Van Heen, our Senior Director of Research and Clinical Strategy. Between them, they have decades of drug development and clinical trial experience. Joining them as presenters are Dr. Amir Zuberi from Jackson Laboratories, Dr. Guoping Feng from MIT, Dr. Chris Hempel from Expressive Neuroscience, Nasha Fitter from Citizen, Dudley Tabakan from VivoSense, and Dr. Bryce Reed from the Center of Health Management at Duke University School of Medicine. Each of the presenters are going to discuss a resource that we have been developing and that will be at your disposal. This is not meant to be a comprehensive discussion of each resource, but rather a brief introduction. And then if you have questions, you can get in touch with us for more information. And we also envision that this will be a first of multiple webinars where we'll keep you up to date on how the resources are developing. The Rett Syndrome Research Trust was launched in 2008, and our focus from the very beginning was on therapeutic strategies to target the root cause of the disease. While Rett syndrome is a terribly debilitating disorder, it does have 3 advantages. It's a monogenic disease caused by alterations in the MECP2 gene. Brain cells don't die. And we know from a 2007 paper from Adrian Bird that if levels of the MECP2 protein are brought back to normal, every symptom measured in the Rett knockout mouse model reverts almost to wild type levels. So our focus is on strategies that target the root cause of the disease. And you can see here the 6 strategies that we've been championing. Now, our business model is to identify these therapeutic areas of interest, to recruit the best scientists that we can in each of those areas, to fund the research, thereby incubating these programs, and then to try to transition them to biopharma. We also identify resource gaps that are important to biopharma, and we try to fill them. And it's really in that spirit that we're coming together today to give you an update on the resources that we are developing in partnership with our presenters. Now, to date, the research that we've been incubating and or through the resources that we have developed, there are 7 companies that have publicly announced a Rett syndrome program that targets the root cause of the disease. We hope to see many more names on this list soon. The next slide shows us the current MECP2 therapeutic pipeline, both academic programs that are in orange and the industry programs listed by each of the 6 therapeutic strategies. The circle represents where we are now, and the arrow is where we hope to be in 2024. Now, this graph is also on our website under the research tab if you want to take a closer look. Now, before I turn things over to our first presenter, I just want to mention very briefly a paper by Christoph Bankowitz that was published on Monday in Nature Communications. It describes a small gene replacement clinical trial for AADC deficiency, a rare genetic disorder caused by deficient synthesis of dopamine and serotonin, and it causes severe developmental disability. The trial treated 7 children, ages 4 to 9, with AAV2 administered directly to the brain in various regions. Not only was the trial safe, but perhaps unexpectedly, the patient's symptoms improved. 6 to 7 regained normal head control, 4 to 7 could sit independently, 2 could walk with some minor support, and autonomic symptoms like oculogyric crisis, where the eyes go upwards and the patient loses control of their eyes. It's also seen in some kids with Rett syndrome. This was resolved in 6 out of 7 of the patients. With continued and intense therapies, who knows how far these children will go. I found this paper just incredibly encouraging as it contradicts the dogma that there are critical periods of development that are set in stone. Now, the Bird mouse model reversal paper from 2007 also contradicted that dogma, but it's so encouraging to now start to see human data. The Rett programs that you are developing or that you're exploring now have the potential to change lives, not only for future newborns diagnosed with Rett at birth, but for so many, hundreds of thousands, like my daughter, who are living with devastating symptoms today. So I want to thank you on behalf of all Rett parents around the world for your incredibly important and urgent work. Patients are waiting, so let's get started. Our first presenter is Dr. Amir Zubairi, Technology and Resource Development Scientist at the Jackson Lab. Go ahead, Amir.
Thank you, Monica, and thank you for the invitation and the opportunity to share with everybody our recent update on the development of new mouse models for Rett syndrome, and also the development of a standardizing phenotyping platform that we think will be very useful for drug discovery and drug efficacy studies. Next slide, please, Jana. So just to quickly outline some of the clinical features of Rett syndrome that we see can be mimicked in the mouse in terms of an equivalent phenotype. So the obvious things like growth can be measured with body size, body weight, various changes in muscle tone, and sort of uncoordinated movements can be looked at as a hypertonic assay, and also with measurements of limb clasping, rotor-rod analysis, and open field studies. Breathing abnormalities which are associated with the clinical syndrome can be somewhat replicated with whole body plasmography and looking at sort of breathing characteristics in the mouse. And seizures can be evaluated also using a sort of electroconvulsive therapy. And collectively in the mouse models, historically, sort of various aspects of the phenotypes have been kind of grouped together and given a numerical score, which is expressed as the BIRD score, which you see on the bottom left there. Next slide, please. So the combination of those assays allowed us to develop a phenotyping platform that can potentially extend up to 24 weeks, where we evaluate on a weekly basis early on BIRD scores, plasmography, and we move on in the later time points to look at coordination and open field studies, leading to a sort of conclusion at the seizures. So now not all mice are going to survive for that length of time, but it allows us to do a comparative analysis of different mutant alleles, and also to look at sort of various drug efficacy studies and effects on subphenotypes. Next slide, please, Anna. So very quickly, I just wanted to update everybody on what the current state of publicly available mouse models for MECP2 are at the Jackson Laboratory, and they can be accessed through the website there on the bottom right. So we have a number of sort of gene deficient alleles, and over on the left, you can see that the stock number 3890 is perhaps one of the most commonly utilized studies in our in vivo pharmacology area, and it's a deletion of both exons 3 and 4 of the gene. We also have preclinical models available on the upper Venn diagram there, and some are associated with reporter models such as TAVI or EGFP. And over on the right, you can see we've got a number of transgenic models which have been made by the scientific community and made available through JAX. And one thing which is apparent here is that we can see, gross defects in mouse phenotypes as a result of even moderate levels of overexpression of MECP2 on the transgenic models 8679 and 8680. Obviously, the higher level of expression gives you the more severe phenotypes. Next slide, please. So one of the challenges we have with that collection that I just showed you is that those individual mutations that were generated independently in different labs, and they're on different genetic backgrounds. To some extent, we do try to standardize in making them congenic, but the strains can contain variable extents of that congenic donor segment around the MECP2 gene on the X chromosome. And there may be unidentified segregation present elsewhere in the genome as well. Some of the constructs are refuse to reporter tags, and we don't really have a good handle on what the effect of the reporter tag itself has on MECP2 function and protein expression levels. And the number of clinical alleles which are available is limited, but growing. And so this makes a direct phenotypic comparison between these strains somewhat challenging, as well as the important sort of correlation of mutation to structure and function. Next slide, please. So with support from the Rett Syndrome Trust and also with additional funding from our NIH-funded grants, we were able to generate an allelic series of MECP2 clinical alleles in the C57 black J background. And so what this cartoon shows you is the intron-exon structure of MECP2 and the relative position of each of the alleles that we've generated using CRISPR-Cas9. The ones in red are the clinical associations. The ones in blue were designed as potential sort of therapeutic exploration areas in terms of what would be the effect of a base editing strategy on, for example, the ARG168X mutation, converting the premature termination codon into a tryptophan. And so we have a collection of 4 of those as well. Now, collectively, those clinical alleles add up to about 60% of the known variants out there in the clinical community. So we think that this is a valuable resource that should hopefully accelerate our understanding of the individual alleles and their biology and potential therapeutic directions also. Next slide, please. So just to quickly conclude, this panel of 9 preclinical models for mutant alleles should be useful to the community. On our standardized phenotyping platform, we're trying to essentially perform what should be a very rigorous platform to address multiple subphenotypes of Rett syndromes that occur clinically, and hopefully that we'll be able to see them in the mouse also. From our early studies in making the models, we certainly know that survival of hemi males is variable and so allele dependent, and we hope that this platform can kind of tease out some of those differences as well. And this phenotyping platform, once it's up and running, should provide a very strong basis for drug efficacy studies on an allele by allele comparison. And that leads to the sort of potential for new drug discovery also. Now, genetic background has a dramatic effect on disease severity, as we know, and clinically, this can be demonstrated as well in the often patients with the same mutations manifest at different levels of the disease. And so with the resources that we've generated and the guides and tools available, we hope that we might be able to very quickly generate an equivalent allelic series on any one of a number of different genetic backgrounds. This is kind of going to be sort of moving in the direction of more of a personalized medicine type of approach. Thank you for your attention, and happy to stay until the end to help answer any questions.
Thank you, Amir. We are going to take questions at the end of the presentations. Feel free at any time to put your questions into the Q&A. Our next speaker is Dr. Guoping Feng. He's the Patras Professor of Neuroscience at the McGovern Institute for Brain Research at MIT and at the Stanley Center for Psychiatric Research at the Broad Institute. Go ahead, Guoping.
Thank you, Monica. So you just heard there are a lot of excellent mouse models from Jackson Lab, and Amir just talked about in great detail. So we got also funded by Rett Syndrome Research Trust to try to fill some gaps especially for genome editing and therapeutic testing. So one thing for genome editing, we realize there are a lot of polynucleotide, single nucleotide differences between the mouse and the human. So if you develop guide RNA from mouse model, then you might have to change it when you into clinic, and it's very difficult to test. So we decided to humanize a region of the mouse that allows to design and testing genome editing, including DNA editing, RNA editing, And if it's effective, then you can directly move the exactly same oligo into testing in human cells and humans. So that's our one purpose, is generate new alleles of mouse models in a humanized background. So we did two things. One is generate wild type mouse with a humanized region, which is the exon part of exon 4, which includes the top 6 of the most common mutations found in Rett syndrome patients based on the database. So I show these slides. These vertical bars are, the top line is the mouse sequence, and the bottom line is the region is the human sequence. This is the vertical wide bar is show the difference between a nucleotide between the mouse and human. The second bar is actually the comparison between marmoset and the human. I will talk about marmoset in a minute. So we basically humanize this region. We generate a wild type mouse line. So this is our control. We want to make sure that humanize this region does not cause a problem. So, and so far these mice have been characterized and work really well, just like wild type C57 mice. And we now use this as a background and actually are generating 3 different mutations. One is already accomplished, R276X, premature stop codon, and we're generating 2 more, which is T158M and R306C. These are all some of the most common mutations found in Rett syndrome patients. We hope these models will be useful, especially for genome editing, DNA and RNA editing, such as so. We will deposit it to the Jackson lab for all these mice. And if someone needs it now, actually it can be obtained from my lab, and it will take a little time to rederive and everything to Jackson labs. So the second goal is to develop a non-human primate model, because there are also many other issues we want to address, like at the circuit level, at the BBB penetration, all different bigger brains. So we now have successfully generated user-based editing R270MX marmoset model. Marmoset basically, they give twins and they give birth twice a year, but their gestation is about 5 months, and their sexual mature takes about 12 to 18 months. So the current the model is only about 6 months old, the oldest one. So it would take a little while before we can have these F1s, next generation, available to everyone. Once they are sexually mature, and we were actually doing IVF to generate many of them at the same time. So they allow us to, test some of the genome editing or therapeutics. And we're hoping to collaborate with the National Primate Center, just send these sperms to them. Hopefully they will be able to distribute to anyone who wants them. So thank you.
Thank you, Working. Okay, my colleague Jana is going to talk about a biorepository that we've developed.
Hi, everyone. So I'm going to talk about our fibroblast and iPSC collections. At the end of 2018, we started a clinical study where we enrolled around 35 families to help us generate these very important resources, not just the biosampling, but we also did some feasibility on some biosensors that we'll discuss a little bit later as well. And so from the families that participated in our biosampling, we had around 31 families. We ended up collecting skin biopsies as well as whole blood from these families, and we were able to generate a repository of about 18 unique mutations. And so these fibroblasts are primary lines that are housed at Rutgers University Cell and DNA Repository. They did the processing of the samples, and that has now become Infinity Biologics. But you can see we have a number of very common mutations listed here in this table. The black text is the protein mutation, and the pink text represents the genomic mutation. And this is a private collection, so they can be ordered through RSRT directly. Now, our iPSCs were also generated from the same group of families. The ones highlighted in pink indicate the iPSCs that are available. We worked with Harvard Stem Cell Institute, their IPS core specifically, to generate these IPS cell lines. And we took great care in generating these. So we used primary lymphocytes as the starting source, and then through Sendai virus, they were created or they were made into iPSCs. And then we took some very careful steps to characterize them. We confirmed the genetic mutation. We did some karyotyping. We looked at embryoid body formation as well. And then we further characterized them with immunohistochemistry to look at trimethylation of histone 3, lysine 27 to look at their exon activation status, which can also indicate erosion over time. And so these are stably exon activated. And we've also quantified their MECP2 expression as another verification of their mutation status. These lines are housed at Coriell Institute for Medical Research, and they're we have a specific page for them actually. So you won't find our fibroblasts or the iPSCs in the standard catalogs for these biorepositories. You can find our cell line list and you can order them directly from us. Our cell line list, if you're interested, are available on our website. You can also find our Rett syndrome collection on the Coriell website on its own page. And then we also have our Add Gene Resources page, which lists these lines as well as some additional iPSCs that are available from some of our academic investigators that are not part of this collection as well. And if you're interested, you can email us directly, either email me at jana@rsrt.org or our standard info@rsrt.org. There's a small or a quick MTA process that's required, and then we can get you the cells relatively quickly. Since we opened these biorepositories, we've had a lot of interest. We have about almost a third of requests have come from industry.
Thank you, Jana. Okay, our next presenter is Chris Hempel, co-founder and CSO of Expressive Neuroscience.
Hi, everyone. Thank you very much, Monica, and Jana, and Randy, for giving us this opportunity to introduce our company to you. We are Expressive Neuroscience. We're a CRO that provides quantitative, high-throughput neurohistology to the gene therapy community. This includes full end-to-end histology services. We generate high-quality immunofluorescence data sets that let us map and quantify expression of proteins of interest right down to the single cell level. And the platform that we've built enables high-throughput acquisition analysis. So we're able to measure hundreds of thousands of cells per animal across large animal cohorts, and to do so at an affordable price point. Next slide, please. So let me just very briefly describe the platform. It consists of 3 components. There's the histology pipeline, ExpertDB, which is the database and analysis engine, and then ExpertWeb, which is the user interface. So the histology pipeline it's fairly standard. We get animals, extract and section brains, stain the sections, and image them. I just wanted to point this out because on the left side of the diagram here, you can see the different points that we can receive input materials from the customer. So we can accept animals, brains, or sections, or we can take care of the entire process from beginning to end. Next slide, please. So we want to use the platform we built to help to contribute to make gene therapy for Rett syndrome a reality. And what we're offering is really about seeing in a clear and comprehensive way where gene therapy payloads are going in the brain, and answering questions like how well is the vector penetrating the brain, how well does its neuroanatomical distribution and cell type distribution match that in healthy individuals, is the level of expression too high or too low, or is it too variable from cell to cell? So it's questions like these that we're set up to answer in a comprehensive and quantitative way. And I hope that there's some groups, people on the line here that are developing gene therapy vectors for Rett, because our services are really geared to helping you optimize those vectors. So in the next couple of slides, I'll give you some examples of the measurements we provide. We typically start by providing a brain-wide mapping of where your gene therapy payload is expressed. So for controls, we can provide a map of MECP2 and wild type sections that's shown here in the left column. And this is, of course, the goal state for any gene replacement vector. By the way, each column here shows sections from 3 different animals of the indicated genotype. We also have disease models that we can provide as controls. So here you're looking at female heterozygotes for loss of function MECP2 mutations, and males completely lacking MECP2 allele, a functional MECP2 allele. And then in the right column, you see the evaluation of a gene therapy candidate that has been systemically injected into knockout males. So you can see that this vector very effectively delivers MECP2 in the brain. Of course, there's been great progress in this field over the last few years. However, the pattern differs substantially from the wild type pattern in a way that may not be desirable for a therapeutic. So based on this kind of assessment, one might decide to stop developing this particular vector class in favor of another one that more faithfully restores the wild type pattern of expression. So we're set up to make this kind of measurement on large cohorts of animals and vectors and help you decide which of your gene therapy development candidates are worth pursuing. Actually, this, can we, Jenna, skip past this slide? Yeah, there we go, thanks. So in addition to brain-wide mapping of expression, we can deliver precise quantification at the cellular level. And here we start with higher resolution data sets. At this level of resolution, it's possible to segment the images into individual cells and measure MECP2 levels. We can also classify the cells using counterstains and measure MECP2 levels in any brain regions of interest. Next slide. So here's an example of this kind of quantification. This shows neuronal MECP2 in 8 animals in layer 5 of neocortex. So each point here represents one cell according to its MECP2 immunofluorescence plotted on the y-axis. And we see that control animals all have roughly the same level of MECP2 on average, and that cell variability is in a fairly tight band. The gene therapy treated knockouts do show a rescue, but again, differs substantially from expression on the in the wild type. Some cells are in the right range, but most cells express little or no MECP2 or much too much. And also there's a big difference between from animal to animal, suggesting potentially dosing issues. So here again, you have a clear metric of vector performance that can be used to rank candidates relative to the goal state represented by the wild type. Next slide. So yeah, just to sum up, I've shown you 2 of the services we provide: brain-wide expression mapping and cellular level quantification. We think these could be very useful in gene therapy programs to guide optimization of things like capsid design, promoter selection, production runs, administration route, and so on. Our standard projects range between 500 and 1,500 per mouse, and that depends on how many sections you want to do, how many brain regions and cell types you want to sample. And then we also do custom work. So again, thanks for the opportunity to introduce ourselves, and we'd be happy to be put in contact with you. Thank you.
Thanks, Chris. Our next speaker is Nasha Fitter. She's VP of Rare Disease at Citizen. She's also a fellow rare disease parent. Her daughter has a FOXG1 mutation. Go ahead, Nasha.
Thank you, Monica. It's great to be here. And we're going to be talking about our partnership with Rett Syndrome Research Trust to build rich longitudinal natural history for 100 females with Rett syndrome. And the way the process works is caregivers come to the platform, they consent. We use the patient's HIPAA right of access to go and collect every single piece of medical information on that patient. So we're not connected to any EMR systems or reliant on Epic. We literally go to the source and we collect all genetic testing reports, neurology notes, imaging studies, etc. We have all of that information. We put it into the patient's account so they have access to it, which is an immediate benefit for the patient and helps us with recruitment. And then on the next slide, as you'll see, we can take those that raw information and actually turn it into something useful. So we've invested in a natural language processing and machine learning platform that extracts information and codifies it. And it's really a partnership with also human assistance. There's quite a bit of it in neurology, and the data output is structured. In essence, we're taking Word and turning it into Excel. And to give you a brief example of this, You'll see on the next slide, here is a typical neurology note, completely unstructured. A doctor has basically written a couple paragraphs, but it's filled with rich information. So I've just circled some of the entities that we would derive from a note of this size. You could see in the second paragraph, for example, decreased tone throughout is circled. The way that we would classify that is generalized hypotonia. Now, physicians across the country and across the world will use various synonyms for the same term. There's 40 different terms just for heart attack. So a big part of what we do is actually capturing these synonyms, and there's hundreds per entities. And as you'll notice next to generalized hypotonia, you'll see that there's a code, and that is the SNOMED code. SNOMED is a globally recognized way to clinically phenotype disease. And it allows us also to submit new codes and get them accepted. So we're continuously building this repository. We also use LOINC for measurements and RxNorm for medications, where we also pull out ingredients within medications. So this is just a quick example to show you how the process is done. We started in our partnership to look at 21 females with Rett syndrome. And I have a couple slides just to show you some of the data that's come out of it. So as you can see, these females were older in age. The average age was almost 18 years, and we were able to pull natural history from about 13 years of data. So it's quite a good chunk of longitudinal data. You can just see the average institutions that a patient visits is about 7. So there's a lot of information in various medical centers that need to be pulled. The documents per patient is over 300 and the pages are over 2,000. So there's a lot of rich data there that we looked at. One of the most important things is getting the genetic findings, the genetic variants, and actually classifying them and then categorizing them. So just in this small sample, we were able to pull out what are some of the common genetic mutations we're seeing within these Rett syndrome females. And this will become much richer when we look at a cohort of 100 and possibly more than that. Clinical phenotypes, we do a lot of work pulling out the phenotypes. And as you can see here, this matches what we know with kind of diagnostic criteria for Rett syndrome. The top 2 being neurology and musculoskeletal with scoliosis and epilepsy. But we also categorize the rest of them, which could be very strong secondary endpoints in a clinical trial. One of the beauties of the system, again, is because we're looking at longitudinal history, we can see changes within these clinical symptoms. So when we looked at neurology exams, we looked at a total of 231 exams, which averaged out to be about 6 per child. You can see in the early years, muscle tone, there's basically a change in muscle tone that we were able to understand and really clearly document through a patient's life. So there's decreased muscle tone in the early years, frequently observed, and it changes in adolescence and adulthood. So again, these kind of measurements and the way that we can chronologically map this can be used for clinical endpoints and also just to see what the natural history of progression is going to be in these females. So I just pulled out a couple examples. We look at the following entities and we really try to capture the entire ontology and the entire kind of picture of what this patient is going through. And just to wrap up in conclusion, we have a strong connection with the community and with these patients, and we give them immediate value, which helps us get them excited. We do all the work of going to all of these medical centers and pulling out. So it's not site-based. It doesn't matter where the patient has been seen. And then our systemic extraction, the idea is for Rett Syndrome Research Trust and all the partners that there's a platform of discovery. You don't need to know the research question you want to ask to be able to analyze the data. And for biopharma, the idea is that this is easily accessible data that allows for genotype-phenotype correlation, endpoint selection, biomarker discovery, and also synthetic control arm in a clinical trial.
Thank you, Nasha. Go ahead, Jana. She's going to talk about a global registry that we're going to be launching very soon.
All right, so when we were trying to decide what we could do to aid biopharma in terms of generating additional data sets that could be useful for clinical development, we realized that there was a lot of sources of untapped data sets that we should probably try to gather. And as as Nasha just mentioned, the Citizen platform is one of those. And so to kind of counter that, we decided we really needed a parent-reported, fully remote, global database that could generate clinical trial grade research-ready data sets. And so we set out to create this resource. And this registry will have a mobile app, so we can ensure that parents have the ability to enter data when it's convenient for them, not just when they're sitting at a computer. And one of the main goals of the registry is to facilitate day-to-day care for their loved ones with Rett syndrome. And so we are planning to provide a number of unique resources for families that can actually improve their child's day-to-day care, but at the same time utilize that data for research purposes. And so some of the information that we're going to be capturing in the registry, it's very comprehensive. We're going to be looking at diagnosis and mutation information. We're looking at symptom burden. We're collecting information on Rett history and developmental history. We're also going to be collecting comorbidities, care strategies, including medications, therapies, where patients are being seen, what types of specialists are on their care teams. We're also going to collect medical events like hospitalizations and surgeries, emergency care use, and also family life information, impacts on parents and work. And we're also capturing perspectives on clinical trials to try to break down barriers for families to participate in different trials. We're also providing families this individualized symptom tracking where they can track symptoms on day-to-day basis or week-to-week basis to help them actually provide their physicians data to help them make clinical decisions. So that aspect of the registry is all parent-reported, fully remote, and will be global. When we're hoping this will generate an extremely broad data set that can be useful for pharma. And our clinical component it will come from the Citizen platform that Nasha just discussed, where we will be able to consolidate their medical care received at any medical provider to help add into this data set. Our registry will also have a dedicated research arm where we can design and conduct remote studies. And some examples that we're planning already, this is a configurable module of our registry where we are planning to validate some questionnaires, do some biosensor development. We can test over-the-counter medications or develop new supplements for GI function. We'll have a research group that if we wanted to do epigenetic studies, collect blood, things like that, we can do all of those things through this platform. And if we wanted to add in some clinical work, we can do that as well with some different configurations. And another main goal of all of this work really is to support clinical development. And our plan is to launch the registry with our pioneer data already entered. So we have a small group, well, it's not that small. We have a group of individuals who have signed up for a pioneer program. Well, they will enter their data prior to our global launch. So as soon as the database is open, it will be immediately valuable from a research perspective. So as I said, one of the main goals is really to facilitate and expedite therapeutic development. And so we've had our hat on for how do we best support clinical program planning and trial design. We want to be able to provide feasibility information, help biopharma select endpoints, validate endpoints, or even create new ones. And the team internally at RSRT, we've had a lot of experience over the years working with rare pediatric CNS disorders, managing and designing clinical trials, working with data, the reporting and all of the documentation that goes along with a high quality data set. We're working towards that. And some of the quality measures that we've implemented based on this background is our database will be 21 CFR Part 11 compliant. We're also coding our medications to WHO drug, and a lot of the standard response options and the comorbidities that families can enter will be MEDRA coded as well to lower level term or preferred terms. And the MEDRA terminology actually can code over to SNOMED, so we will have an ability to integrate the citizen data as well. We're also going to collect genetic reports so we can source verify the mutations that are reported by parents, and we're working with a certified genetic counselor to do that. And then we're also going to be providing the database in certified translations so families can enter their data globally in their native language. And we're also working with FDA to maximize how our registry can support therapeutic development to make sure that we maximize this opportunity for biopharma. And in addition to just what, our internal team, we have a bunch of different external groups we're working with, our clinical leads and clinical advisors, which you probably know well already, Bernard Suter and Carrie Fu, Liz Berry-Kravis, and Eric Marsh. We've also been working with a wonderful parent committee. We have been leaning on a lot of advocacy groups who have already had successful registries. We're working with a psychometric analyst to help us with the language choices and the determination of how to assess symptom impact, and our genetic counselor, like I mentioned. And then we also have a really great biopharma advisory committee that's been weighing in and helping us design this database to meet the needs of groups that have active or planning active Rett programs.
Thank you, Jana. As you can imagine, this is a heavy lift project, and we're looking forward to getting started and starting to generate some data. Okay, our next speaker is Dudley Tabakin from VivoSense. Go ahead, Dudley.
Thanks, Monica. Yes, Dudley Tabakin. Today I'm going to speak today about outcome measures, digital outcome measures. VivoSense is, we specialize in agile end-to-end solutions for digital outcome measures from wearable sensors in clinical trials. So when we talk about a digital clinical measure, I'm talking about anything that can come from a wearable sensor, such as your wrist-worn actigraphy device or an ECG patch, or anything that you can wear at home on the road, even a weight scale and so on. So why would you use a digital wearable clinical measure in Rett syndrome? There's a number of very good reasons. I'll just list through them here. Novel endpoints that previously may not have been measurable in the clinic and may be more sensitive to treatment effects are possible. Your recruitment reach, you can expand to out of the clinic and measure real world evidence at home. Objective endpoints can replace or supplement existing subjective measures that are being recorded in a patient diary or electronic patient recorded outcomes. You're looking at real world measures, which may better represent a meaningful aspect of health to the patient and caregiver. For example, looking at their activity over the day versus their activity in the clinic. A wearable sensor also gives you the opportunity to collect more than one endpoint at a time. Heart rate plus activity can be collected at the same time with a single sensor or multiple sensors. And then we can provide context from the data from a patient recorded outcome to an objective measure from a sensor. And we allow for continuous assessments and rigorous investigation to the timing of events and treatment effect of any therapeutic. Next slide, Jana. So with RSRT, the VivoSense team have been working on a number of studies, particularly on respiration, apnea detection, abnormal breathing in the population. Polysomnography, which is a gold standard for sleep measurement, has been used in Rett syndrome studies for many years to detect apneas, hypopneas, change strokes breathing. And one of the pieces that's used most often in identifying those is the respiratory inductance plethysmography, or the two bands, one around the chest and one around the abdomen, which measure the chest and abdominal wall movement. And from that can identify apneas, hypopneas, tachypneas, and change strokes breathing. So we have been using that in a number of studies, and I will take you to a study that we have been doing with RSRT, and this is the VivoSense software platform that you're looking at, and we can readily see apneas and hypopneas being identified here. The top chart is the tidal volume measured from derived from the abdominal and rib cage bands in a respiratory plethysmography. The respiratory rate is in red and the timing of each breath in green, and we can see how apneas are easily detectable from the respiratory plethysmography data in the patients. Next slide, Jana. So working with the RSRT team, we've also been looking at novel methods to measure abnormal breathing, apneas, and respiratory rates. Two methods that we're working on right now, ECG-derived respiration. So using an ECG patch that records ECG signals, we can determine the R wave to R wave interval timing. And from that, understand the apneas and respiratory rates, or derive apneas and respiratory rates within the data. This allows for more continuous measurement, multi-day measurements. ECG patches can be worn for 14 days versus respiratory plethysmography, which typically, if you wear that for 4 hours a day, is more than enough for any patient or healthy person for that matter. Another novel method we're looking into is actigraphy-derived respiration. So by using actigraphy devices or accelerometers on the chest and abdomen and looking at the chest and abdominal wall movement patterns, there's the potential to once again derive or understand measures of apneas and respiratory rate because of the low activity typically in the Rett syndrome patients. I'm just going to, sorry, Jana, can you stay on that slide just one more moment? I wanted to add that there's other actigraphy measures from wrist-worn devices or ankle-worn devices that would measure the movement and activity, behavioral activity of Rett syndrome patients, which are also interesting and novel ideas that we have been investigating. Okay, next slide. So this slide is once again back in our VivaSense software. And what we've done here is taken on the top chart, the tidal volume from respiratory plethysmography and compared that to ECG-derived respiration from a heart rate monitor, ECG patch. And we can see, so the top chart is the tidal volume from respiratory plethysmography. The red is the heart rate. The blue is the respiratory pattern derived from the ECG. And at the bottom in green and blue, we have in green the time of inspiration for each breath from the respiratory plethysmography, and in blue the time of inspiration from the ECG. And we can see a good match between the two. So ability to use the ECG patches to identify apneas in the respiratory pattern, which will be very useful for long-term continuous study of Rett syndrome patients. Okay. And to conclude, the use of digital sensors in Rett syndrome is going to be groundbreaking, in our opinion. Digital sensors can provide novel outcome measures, as we've seen already a couple there. Digital sensors are typically low patient burden, so you can use them at home. The respiratory plethysmography nowadays comes in a vest. You could use an ECG patch, or if you wanted to go even low patient burden, a wrist-worn actigraphy could provide certain information that might be useful as well. We can provide insights into meaningful aspects of patient health and caregiver concern. As an example, if the caregiver concern is lack of sleep, we can look at the meaningful aspect of that. How do we improve sleep in these patients, or is the therapeutic improving sleep in these patients by measuring their sleep, for example? And then potential to provide measures of patient behavioral activity. How often do they get up from their get up and take a walk and do things that are typical in everyday life? And continuous monitoring enables identification of transient events like we saw in apneas, and the timing of these events. Do they happen more often in the afternoon, in the morning, at night, and so on? And that's it. Thank you.
Thank you, Dudley. Okay, our next speaker is Bryce Reeve. He's the director of the Center for Health Measurement at Duke University School of Medicine. And he's going to be speaking about a communication assessment tool that we are exploring for Rett syndrome. Go ahead, Bryce.
Thank you so much, Monica, and thank you to you, Jana and Randy, for allowing me to speak to you all today. So I'll be talking briefly about the, previous slide. Thanks. About the observer-reported communication ability, or ORCA measure, which is a caregiver or parent-reported measure or questionnaire survey that is an assessment from the parent's perspective of their child's communication ability. And this measure is designed for use in neurodevelopmental disorders that may be used either for observational studies, natural history studies, or clinical trials. Next slide, please. So for full disclosures, I am one of the developers of the ORCA, and therefore I need to disclose my attachment to the ORCA. The ORCA is currently available to the public and for nonprofits and academics. The measure is free to use in the public domain. However, for commercial entities, there is a fee and a license required to use the ORCA measure. So as brief background on the ORCA, there's lots of different neurodevelopmental disorders. And each one might be deemed rare, but collectively it represents a significant public health need. And importantly, as we observed in the previous presentations during this webinar, there are many exciting new treatments and interventions which are promising to have impact on addressing symptoms or potentially improving the lives of children and individuals with Rett syndrome. And so importantly, what we need to make sure as we do these trials in humans is that we're assessing outcomes or treatment benefit that are important to those with Rett, as of course, as with the parents and caregivers, what we call patient-centered outcomes. So importantly, as we get ready for these trials in humans, we need to have well-qualified, validated, and meaningful outcome measures. And so communication ability has been identified both in Rett and other neurodevelopmental disorders as a high priority area for which parents want to see improvement in their children with Rett or other types of conditions. Next slide, please. Therefore, we developed the ORCA measure, or Observer-Reported Communication Ability measure. And the ORCA measure assesses a broad range of communication ability, including expressive, receptive, and pragmatic forms of communication. The measure is targeted towards individuals with significant deficits or limitations of communication ability, and often these populations are typically nonverbal. And importantly, the ORCA measure we recognize that children communicate in a number of different ways, which could include, but not limited to, gestures, sounds, vocalizations, or using an assistive device. The ORCA measure is designed specifically from the parent or caregiver perspective to complete the ORCA independently without the assistance of some type of administrator. And typically it takes a parent about 15 to 20 minutes to complete the full ORCA measure. Purposely, we have designed the ORCA measure for use in regulatory trials following FDA guidelines for how to develop and evaluate clinical outcome assessments like the ORCA measure. Our original development of the ORCA measure was specifically for the disorder called Angelman syndrome, but we designed it in a way that it could be applied to other neurodevelopmental disorders, just like Rett syndrome itself. Next slide, please. I will not go through all the points on this slide there, but importantly, it points out our process. We follow a rigorous scientific process consistent with FDA guidelines to develop and evaluate the ORCA measure, which includes both formative stages, qualitative phases, as well as psychometric or quantitative phases to make sure what we're developing is relevant, reliable, and a valid reflection of communication ability from the parent's perspective. Next slide, please. The ORCA measure is currently made available and licensed through Pattern Health, which is sort of a subcontractor to Duke University, and there's the website. It is available both in English and in other languages such as French, Dutch, Spanish, and Italian. And through a contract with the Rett Syndrome Research Trust, we're in the process now over this next year to conduct both qualitative and quantitative approaches to validate the ORCA measure for use in Rett syndrome. Next slide, please. And with that, I conclude by showing our awesome team. On the top row are methodologists. These are experts in the evaluation and qualitative and quantitative methods to develop the ORCA. And our bottom half of the screen are the clinicians that are speech language pathologists and speech development experts that help guide and develop the ORCA system. And with that, thank you very much. Monica?
Thank you, Bryce. I'm going to just talk very briefly about the Rett Working Group. You go to the next slide, Jana. This is a series of virtual meetings that the RSRT is hosting together with Fyodor Yurnov, who's the scientific director at the Innovative Genomics Institute. The meetings happen every 3 weeks or so. And each one has a topic, a specific topic. We have two speakers for each session and a panel discussion. And the goal is really to develop a roadmap to IND, which is therapeutic agnostic. We have had 9 sessions so far. All of them are recorded and available. They're password protected, but if you are interested in joining these sessions, please email me, and I can also give you access to the recordings that we have so far. Our next session, we're going to be giving an overview on preclinical research, and then we move on to our clinical sessions. It's been a really interesting program that we're doing, and I hope that the publication, which hopefully will be published in a high-profile journal, will prove helpful to everyone. Okay, Randy unfortunately found himself on the road today, kind of unexpectedly. So I'm going to talk briefly about how RSRT works with biopharma. So we're here to help you, right? Your success means success for all of our patients and families who we represent. And so we're here to help you in any way we can to be successful. We strive for transparent and collaborative interactions. We help you identify resources if the resources don't exist. We try to generate them in support of your programs. We guarantee absolute confidentiality. We're trying to aggregate and share pre-competitive ideas and knowledge. And we're also starting to engage, educate, and collaborate with the FDA. If there's something that you need that we haven't covered, please get in touch. We'll do our very best to help address your needs. Go ahead, Jana.
All right, so I'm just going to wrap up here quickly and just give everyone just a quick overview of all of the things that we discussed today. And this is kind of a timeline for when we feel these resources should hopefully be available to help your clinical development planning. So all of the things we discussed today are ongoing right now. We've got the different mouse models that will be available soon from Jackson Labs and our academic partners. The marmoset is on here as well, which should hopefully be available later in 2023. The Rett Working Group publication of the Roadmap to IND, we're hoping to have that published in Q1 of next year. All of our outcome measures that Dudley spoke about, the Hexoskin, MC10 endpoint, and Emerald that we didn't go into detail. These are all digital endpoints that are ongoing now, and these studies are ongoing now, and we're hoping to have information around their utility as a clinical trial endpoint, whether or not they can detect treatment effects. That information we're hoping to publish from our clinical trials that are ongoing now early next year. The Rett Orca, as Bryce mentioned, is ongoing now. That will be a study that is conducted in 270 Rett families. That new scale specifically for communication in Rett syndrome should be available early next year. And then for our real world evidence, the Citizen Digital Natural History Study and the Rett Syndrome Global Registry, those will be starting. We're still developing the Rett Syndrome Global Registry, but we're planning to have our initial natural history study with 100 patients ready by mid next year. We do have the 20 patient data set available now, if that's of interest to anyone. And once we have our initial database open for the Rett Syndrome Global Registry. I think the first time we'll have data that'll be available to biopharma will probably be Q1 of next year, and then we will continue to collect longitudinal data over time. And so we're hoping that all of these different resources that we're developing, that these timelines will help you in your clinical planning. I'll stop there, Monica.
Okay, great, thank you. Maybe you can stop sharing. Oh yeah, actually that's good. Leave it here so people can get our contact information. All right, we're gonna, there's a couple of questions, we'll address those. I'll also mention that I will email everyone that's registered with a link to the recording for the webinar so that you can watch it again if need be, or share it with colleagues. Okay, here's a question from Andrew Mulberg, and it's directed to you, Jana. How will data duplication be monitored and avoided across two registry efforts?
So we're designing our registry to be independent and separate from the existing natural history study. And our intent is specifically not to overlap. There are certain key data points like genetic information that we need to have for biopharma, for these data sets to be useful for biopharma. So there will be some cases where there will be duplication, but the impact on families will be minimized. So for example, we're purposely not collecting clinical measures in our registry. It's all parent based. So that's where we're starting, and we will expand the registry as needed to fill new gaps as they become apparent.
Great. Here's a question for Bryce from Gopi Shankar. Has the ORCA been used in an interventional study yet? What has the experience with FDA been in using ORCA as an endpoint in interventional studies?
Thank you so much, Gopi, for that wonderful question. And so the original organization that funded our work developing ORCA, the Foundation for Angelman Syndrome Therapeutics, FAST, they did have a early phase one, phase two trial that started last year, which did include the ORCA measure and collected data on it. As you know about the sort of process working with the FDA, is that there's sort of a series of back and forth iterations and meetings with them as they look to evaluate and ask questions about both the outcomes selected as well as the measures. And so we're sort of in that sort of bounce back and forth where we have built like our dossiers that provide the qualitative and quantitative evidence and are feeding about. So we haven't had the final decision on that particular trial yet about their thumbs up or thumbs down. But so far we've heard only encouraging words from the FDA. In addition to that trial, there are a few other clinical trials, many of them in Angelman syndrome, because again, that was our primary focus of ORCA, was in Angelman syndrome. So there are a few other trials that are currently using the ORCA measure. In addition to that, we've actually received quite a lot of interest from other neurodevelopmental disorders to use the ORCA measure. So ongoing in a lot of registry studies and natural history studies, they're starting to use ORCA and collect it in other disorders. In addition, there's other clinical trials. The last thing I'll say, then I'll be quiet, is that, again, we have followed the process that the FDA recommends for developing a observer report outcome measure or clinical outcome assessment measure. Just recently, the FDA funded our group to expand the ORCA measure to a broad range of neurodevelopmental diseases as part of their patient-focused drug development program focused on core outcome sets. So we know the FDA is very interested in ORCA. They've funded us to do additional work and are working collaboratively with us to make sure that ORCA is available to many other broad conditions. And as I know, we are working specifically with the Rett Syndrome Research Trust to make sure that the ORCA is a valid reflection of communication ability in their important population.
Thank you, Bryce. Another question from Andrew. Has RSRT considered a qualification path through FDA to make these outcome measures most useful to all partners, and especially for a validated tool like, for example, the ORCA? Jana or Randy, either one of you want to answer that.
I'll step in since Randy's indisposed. So yes, so we have considered that. We have contacts with FDA. We have already met with them to talk about some of our digital biosensors. We will be meeting with them to talk about the registry. The ORCA is another thing that we will be discussing with them. So yes, we are planning to try to develop these different resources to the best of our ability to make them as useful for pharma as possible. However, there may be cases where we can partner with pharma to help get some of these tools validated through the clinical development path. If we can use some of these devices in the ORCA in clinical trials that are ongoing under an IND, that may possibly help us validate them as we go through. So that's also our plan. We may not necessarily just try to validate them independently, but in a partnership with pharma.
Great. Question from Mitra to Dudley. Do any of the biosensors have the ability to evaluate hand function or movement in Rett patients?
Thanks, Mitra. That's it's a difficult ask for a actigraphy or accelerometer device to measure hand movement in Rett syndrome because of the frequency. The hand movement is measuring accelerometer data or actigraphy data is typically it's measuring the noise that you create when you move your arm anyway. So it's very hard to distinguish between the two. So that has not been done yet, at least not from what we've seen. However, it is possible to measure activity, just general activity, such as, well, if the patient is ambulatory, certainly you can measure the activity during the day. How often are they sedentary, seated, and how often are they standing and walking and things like that. But actually measuring the hand movement effectively, I think that there's a high amount of variability and would be a difficult ask. That's not to say it's not possible. We just haven't tried it yet.
There are some efforts underway to measure hand function with video recordings and to have that assessed with blinded scorers. There was a paper that was just published on that. Jenny Downs, I think, is first author or senior author. You can contact me if you like. We can send you the paper. And it's also something that we're going to be covering, the hand function, something that we're going to be covering in one of the future Rett Working Group sessions. Jana, do you want to say anything about Emerald that might be able to pick up hand function? And it's a pilot study we're doing. It's a small study. We don't have any data yet, but.
Yeah, so the Emerald is an invisible technology. It's not a wearable device, which are the ones that we talked about today. But the Emerald can detect physiologic function from a distance. So through radio waves, it can actually assess an individual with Rett syndrome. And we're piloting that device right now to see if this is a technology that could potentially be used to assess Rett syndrome function. It's possible that we could, it could pick up hand function or repetitive hand motions. We're going to have to wait and see what kind of data we get out of this initial analysis, and then we can decide, if it generates a good data set and it's easy for families to use, and we can potentially look at things like that, but we don't know right now.
Okay. If any other questions come up as you think about this presentation, please feel free to get in touch. We thank you very much for joining. I will email you the link to the recording, and we look forward to updating you in the future as this work progresses. Thank you to all of our presenters, to my colleagues. Oh, let's see, we have one more. Oh, that was a thank you. Okay. Okay, we'll be in touch. Thank you so much. Have a great rest of the week.